begin by talking a bit about your work on selenium, which is the micronutrient most commonly found in Brazil nuts and some types of fish as well. You've done a lot of extensive work on it, and you've documented that in some of your papers where you talk about, the history of selenium and you talk about the good, the bad, and the ugly. I loved that title of that paper, by the way. And you also show how, selenium plays this dual role in aging where it is protective against cancer and also has other benefits like immune function benefits, but at the same time, it can also promote cancer. So maybe if you can talk to us a bit about what is the role of selenium in aging and why is it such an important micronutrient?
Yeah, it's a big topic. So actually I've been interested in selenium since the time I was a postdoc working at NIH, and when I started my own lab, the first question that we asked is how many selenoproteins are present or encoded in the genome, like in the human genome, because at that point only a few proteins were known, and there was an attempt to kind of explain the role of selenium through these proteins. And selenium, of course, is an essential trace element. So we absolutely need selenium. If we would not have it, we would all die. So so we developed this a program to identify these genes computationally. And because selenium is present in proteins in the form of selenocysteine, which is the amino acid, and it's inserted co-translationally, meaning that it's inserted during protein synthesis. So it has its own codon. It's actually we call it the 21st amino acid. So in many textbooks would say that there are 20 amino acids in proteins, but in fact, selenocysteine is the 21st. It has its own codon, it has a tRNA, and it has the machinery for biosynthesis and insertion. And what is needed is the particular RNA structure in the 3' UTR. When this structure is present, then a codon UGA, which is normally a stop signal, is recoded and instead inserts selenocysteine. So what we did, we developed a computational algorithm to identify such structures in the genome. We couldn't search them for like sequences or particular motifs because they are just not conserved. So we had to look for like particular stems and loops, calculate free energy for various structures, and find conserved elements across multiple species. This way we were able to identify the structures in the genome. And we found that there are 25 genes. And this was 20 years ago that we found it. And to this day, actually, it's still 25. So it seems like we haven't missed a single one, and we also had no false positives. And so these genes kind of explain why selenium is needed, because selenium only works through these 25 genes.
So then the converse is also true where these 25 genes are dependent on selenium.
Yes, yeah. And, the functions are, for example, some work in antioxidant defense, like glutathione peroxidases or thyroxine reductases or methanyl sulfoxide reductases. Some work in functions like thyroid hormone activation and inactivation, like this so-called deiodinases. So they catalyze reductive deiodination of thyroid hormone. And there are other functions that are known. So basically, but the major thing is that it's redox. Typically, selenocysteine is just one amino acid. It's always in the enzyme active site and serves some kind of catalytic redox active function. And so this kind of explains why we need selenium. Several such proteins are essential, like a couple of thyroxine reductases. There is a very important protein called GPX4, which is also known as phospholipid hydroperoxide glutathione peroxidase. And this protein is needed for protection against ferroptosis. It's a new type of programmed cell death. It's now a very, very active area of research. There's a huge explosion. And that's a key protein in that. It's actually dependent on selenium. So all this kind of makes selenium really exciting. But then when we think about the role of selenium in aging, so I view it as like a prototypic kind of a feature to kind of examine aging, because it has both positive and negative sides. So it's needed for proteins to maintain, several essential functions and some less essential, but at the same time, it's also toxic. So actually, the body sees selenium as a toxic element, and 90% of it is being kind of methylated and converted to acylated sugar and excreted, and only maybe 10% is actually used for essential functions. So, and this damage due to toxic side of selenium can accumulate over time. So you have this, you, from the aging perspective, kind of antagonistic pleiotropic function with like, okay, is essential important function, but then also kind of negative functions being that contribution of cumulative damage over time. So, and in fact, this idea of biology in having an inherent antagonistic pleiotropic function is something that we proposed some time ago, because this idea of antagonistic pleiotropy is a central kind of evolutionary theory of aging, right? But it primarily operates by alleles, so maybe genes, saying, okay, there are some genes that may be beneficial early in life, but then detrimental late in life, and because they are beneficial early in life, you cannot really get rid of them, even if they contribute to damaging functions. But what we are saying is that these genes and alleles, they do not evolve. They are actually already, every gene has that function, you see, from the very beginning. So these genes cannot evolve because they already, like, it's the basis of life. Not only genes. I give you the example of selenium. It applies exactly the same concept of the antagonistic pleiotropy. So we think antagonistic pleiotropy is a feature that may actually may influence changes in lifespan, but does not really define aging per se.
Mm-hmm.
You see, because it's there from the beginning. It cannot evolve. And so if we take an organism that does not age, like it's a certain, like a simple, maybe unicellular organism that symmetrically divides, for example, uh, such genes are already there. So, but at the same time, aging evolves when we have certain, um, decisions on the organization of the organism, I would say. So, for example, in mammals, we develop um, non-renewable cells and structures like neurons. So they develop during embryonic development, and then they are irreplaceable. So we don't replace them. And so once we have that, sooner or later, neurons one by one would die, right? And we cannot replace. Therefore, aging already must happen. Yeah, in a way. Or if you have, like, at the molecular level, let's say long-lived proteins. So there are some proteins which basically live the whole life; they just not replaced. Yeah, some nuclear pore proteins. There are some other proteins. Obviously, it's a chemical molecule. So when in the harsh environment, or even not so harsh, but still not ideal environment of the cell, they would be modified or broken sometimes. So there would be some kind of dysfunction accumulate with them.
Mm-hmm.
And so aging is unavoidable for that reason only. Just that reason is enough. So we don't need to invoke, like, to evolve aging, this kind of new antagonistic pleiotropy kind of alleles. Yeah, even though they contribute still, once they we have this aging trajectory, of course we could make it faster or slower, and so on.
So what you're saying is, so antagonistic pleiotropy, as you explained, is mostly the concept that something that is good for you when you're younger is turns out to be harmful when you're older. But what you're saying is that doesn't really explain aging, because that's just it's just happening. It's like we've evolved to age. So the pleiotropy theory doesn't explain aging. Is that how I understand it?
Okay, so antagonistic pleiotropy is an inherent feature of life. Okay,
Mm-hmm.
it's there. So we have, let's say, genes or whatever other elements of life, selenium or oxygen, and so we need oxygen for respiration. We need selenium for for some functions, but they also contribute to damaging to cumulative damage. So oxygen, partially reduced oxygen intermediates, so-called reactive oxygen species, they can damage molecules, and this damage may accumulate. Selenium in the same way, it's highly reactive. It also produces
Hmm.
species, other damaging species, which accumulate. So we have this positive feature for any biological feature which we use in life. We have this essential beneficial function. That's exactly what antagonistic pleiotropi say. Okay, this is beneficial allele. That's exactly. It's not just allele. It's any element of biology that we need, selenium and oxygen included, and but also they contribute to cumulative damage, which is kind of the what people say on the when they discuss antagonistic pleiotropi, something which is deleterious later in life. But actually, it's not just late in life. It just simply contributes to cumulative damage. That's all. And
I
it's
see. Okay.
just we detect it and we visualize it and we discuss it late in life, but actually it's a gradual process.
I see. Okay, yeah. So it's just building up over time. So it's like if you have too much of selenium right now, sure, maybe when you're younger, you might not actually see the negative effects, but the damage is building up over time, which you're going to see much later in life,
Yeah.
probably.
Mhm.
Yeah.
Yeah. So that's why I mean it's not selenium. I have kind of two sides. We work on selenium, and of course we also work a little bit selenium and aging, but actually it's a small component
Mm.
of what we work. So we actually essentially almost no overlap. Yeah.
Okay. Because you have documented, though, how selenium helps in preventing against cancer, though.
Yeah.
What
Yeah.
is the connection between selenium and
Yeah,
cancer?
yeah. Well, it's exactly the dual side. So you have beneficial and damaging exactly for that same reason. So you have genes which uh, contribute to which protect against cancer. Let's say thyrodoxine reductase is actually is needed to maintain the redox state of thyrodoxine, and thyrodoxine is needed to support the reduced state of cysteines in the cell. Yeah, because thiols need to be reduced, but then you generate cancer cells and become dependent on thyrodoxine reductase because it's needed even more. So okay, what do? Do you inhibit thyrodoxine reductase? It's just kind of has dual side. Or anyway, so in some contexts, selenium would be protective against cancer, and some actually would be actually promoting cancer.
Okay.
A little controversial from that perspective. I think we could find some context in which it would be actually useful, and then many people work on that. But again, so we need to realize, and it's not only for selenium, but for many other essential features, it depends on the context, depends on the model, on cancer, on tissue, and so on.
Hmm. And is selenium something our body makes naturally, or do we only get it from dietary sources?
Well, it's a chemical element, so we cannot really make it, but it's from natural sources, and it's everywhere where sulfur is. So sulfur pathways, they cannot distinguish between sulfur and selenium.
Mm.
So in every sulfur site, like cysteine or methionine or iron supercluster and so on, there is a little bit of selenium. So, but then again, so selenium is also used very specifically. So the cells somehow recognize selenium and convert it to selenocysteine, which is built on the tRNA, chemically built on the tRNA inside the cells, and then is inserted into proteins.
Okay.
Yeah.
So, from a practical perspective, though, and do you think it's absolutely critical for someone to be getting the recommended dose of selenium, and should they be quite cautious about not getting too much of it?
I would be cautious not to get too much for sure,
Okay.
uh, because there is the level of selenium. So it's not like you get more selenium and you express more selenoproteins. There is saturation, yeah, because you're limited by the machinery. So whatever the expression of the machinery, so under selenium deficiency conditions, the levels of selenoproteins become very low. Yeah, but even then, actually, there is a priority for selenium supplies. Some tissues and organs have priority, like the brain. So even under extreme selenium deficiency, brain levels of selenium are almost normal. Brain and testes has the highest priority, but in the liver we may lose maybe 99% of selenium. And within each tissue also there is priority. Under selenium deficiency, certain proteins are expressed more, and less important proteins are less expressed. There's a mechanism like that. Yeah.
Hmm.
But then when you increase selenium, you get more and more again in a priority like manner, more and more selenoproteins, and then there's saturation. So if we get as humans, but maybe about 55 micrograms of selenium a day, we don't increase selenoprotein expression anymore in any tissue, and instead selenium just contributes to damage more or less.
Oh, wow.
So, uh, especially in the US, we rarely have actually we probably don't have selenium deficiency because the level of selenium in the soil is pretty good, and we eat all kinds of products which shipped from everywhere. So we have typically normal levels of selenium. In Europe, it's a little bit lower. So in some actually countries have a program to supplement foods with selenium, like Finland.
Hmm.
Also, New Zealand has a low selenium in the soil. Uh, but again, so in the US, I don't take selenium. I think we have enough. And in
I
fact, it's
see.
worse to take selenium more than is needed.
Interesting. Yeah, because I typically eat like two Brazil nuts every day to get my selenium, but maybe I need to actually measure that and tone that down.
Yeah, you could measure.
Fantastic. You mentioned redox biology a little bit in its connection and reactive oxygen species and its connected with connection with selenium. Um, do reactive oxygen species still contribute to aging, or is that a myth? Because I believe reading somewhere, maybe I think it was David Sinclair's Lifespan, where he was talking about, antioxidants don't really help. Maybe it wasn't to do with reactive oxygen species, and more to do with antioxidants. But maybe, I guess, talk a little bit about reactive oxygen species and its connection with aging.
Yeah. So we started working with selenium as we discussed, and then we realized these selenoproteins, they are all auxiliary reductases. So I ended up being a redox biologist. So I have this title, director of the Center for Redox Medicine here. Uh, so kind of redox really interests me a lot. Yeah. So and yet I've written this paper with the title. What was the title? That the free radical theory of aging is dead. Long
Hmm.
damage, kind of the damage ideas. And the reason is that uh, the question that we need to ask: can it be a major kind of damage form that defines the aging process? Be it, I don't know, you know, protein damage, or DNA damage, or mitochondrial dysfunction, or or reactive oxygen species. I think not, because if there is a one particular damage form that is most important, then selection would be relaxed on other systems that may produce other damage forms, because they would be irrelevant, right? So because this major form already causes aging. And so in the end, the relative contribution or importance of other damage forms, they would somehow synchronize, yeah, with this
Hmm.
initially main damage form. So we would not have it. That's why all these theories, they kind of failed over the years, because there was a, telomere shortening, or, oxidative damage theory, or, this Leslie Orgel's idea of kind of catastrophe at the level of transcriptome and the proteome because of the inaccuracy in protein synthesis. Uh, so I think a challenge is really how can we, um, test or investigate the entirety of damage. Yeah, not just any particular. Because I think there've been 60 years of research on manipulating genes. Okay, let's remove, knock down a gene for some antioxidant genes. Let's overexpress it, and it led to nowhere. I think it's very clear that it would not. We could manipulate, and in some context, maybe shortly it's conditions. If there's some other damage form, is like really becomes a major, we can rescue it, and yes, with lifespan will be used. But in terms of like conceptual understanding of aging, I don't think we can make progress by applying these approaches. So instead, we need to focus on the totality of damage. But it's very difficult to do. How would you measure it? So we are big proponents of omics approaches, because at least they become closer to that goal. So we could, for example, do DNA methylation and use machinery clocks, try to identify which components of these clocks represent the actual damage in the epigenome. In parallel, maybe do transcriptomic analysis on parallel, maybe metabolomic, or maybe some other forms, and link them with kind of physiological measures. So that, I think, would be more appropriate to study aging. So But perhaps even more important point here is that we really need to focus on fundamental basis of aging. So some people think, okay, we don't need to understand aging. We already understand it. We just need to find kind of interventions. To me, it's like alchemistry. It's like alchemists. That's exactly what they try to do. They take one like element and try to make gold from it, by trying. And that's what we are doing in the field. We are trying all kinds of different interventions, whatever, gene manipulations, without conceptual understanding of aging.
Hmm.
And it's almost completely missing. It's very difficult to fund. It's very difficult to do research, to publish the papers even, because you need mechanism. So you submit the paper to any big journal, and there is a reviewer says, okay, where's the mechanism? But how can we study mechanism? It's just too difficult. The aging is more difficult than life. If you try to understand metabolism of life, aging is more difficult. And and then you try to to, to to use some tricks, like, okay, you find some gene and describe it, or mitochondrial dysfunction, DNA damage, and then you publish a paper. That's it. So it's a it's
Yeah, it's fascinating actually. In all my conversations, there are some scientists who think that we should understand aging better and understand the basic science, whereas others think that it doesn't really matter how we think about aging or define aging or understand it, as long as we can figure out interventions. And it sounds like you're in the camp where you're saying that we should understand aging, and I'm probably in that camp too, but I'm far from the research to comment on that. But my question is then, how do you think about aging? And what do you propose then? How should we be studying it to understand it better?
my thinking. Aging is damage. Yeah. So damage in a broader sense. Yeah. So we have biology. We have a basically programmed biology. The biology is programmed to make a fit organism. Yeah, that can compete with other species and within other individuals of the same species, uh, and programmed in terms of how things develop, how they are maintained. That's what we need genes for. But unavoidable consequence of that is kind of negative side, as we discussed, in terms of the negative consequence of these genes and other functions. And these consequences, they accumulate if we have a particular organization, biological organization of the organism. And that's the damage, and then that's the aging in my mind. Yeah. So when we follow age-related changes, uh, they include all kinds of changes. So they include something which just changes for some reason which is completely irrelevant, doesn't make things better or worse. There are changes which is damage, of course, and makes it worse. And there are changes which are kind of um, in a way protective changes, which the organism finds each time it finds new homeostatic state, and uh, because damage accumulates, organism responds, accumulates, responds. Each time it's new. Yeah, the whole system is new. Yeah, and
Mhm.
so when we try to uh, let's say slow down this process or maybe reverse it, so what should we target? What exactly is our enemy here? So, and that's exactly the question: is what is aging? Because some people understand it differently. Some people say, "Oh, it's just age-related changes. That's aging," and some will say, "No, it's a process which is something which after development, and then the trajectory continues. That's aging." And some people say, "No, it's increased chance of dying. If the let's say Nick Moret doesn't show increased mortality with age, it's not an aging anymore." And some other people say, no, it's dysfunction that accumulates. It needs to be physiological. And so what is it then? Is it like damage, clog, each of the changes, dysfunction, continuation of development, whatever? What is it? I mean, they are kind of similar in some way. They're partially overlapping, but strictly speaking, they're not the same. So in my mind, we need to define what we need to target. Because in some cases, maybe uh, we target mortality, and mortality would not increase with age, but in my mind, organisms would still age. Yeah, like Nick Moret. Or for example, some fishes, their fishes or turtles, or trees, they grow. Let's say tree, uh, it with age, it's often less likely to die because it becomes bigger, like a
Hmm.
wind or some kind of animals cannot really damage it. Also, its fecundity increases because it's simply bigger. there's more offspring it can give. Uh, and so based on this kind of evolutionary principles, we say, oh, it becomes younger. But you look at the tree, you say, no, it's an old tree. It's not a young tree. Yeah. So, okay, there's a contradiction here. And it's the same way. That's why. But in a way, that tree, I think you can see it's older tree and it has more damage, but it's less likely to die, and it has better fecundity. So that's kind of my thinking. So again, so I don't know if I'm clear or not. To me, aging is damage.
Uh-huh.
So that's something we need to track. That's something we need to slow down. Something we need to reverse, even though it's very difficult. We don't need to reverse, some certain genes. Actually, most of the like gene expression changes, and to some degree also even epigenetic changes, they are actually protective. They
Hmm.
can still track the aging process. They still can be used to build clocks, but they do not represent aging. So clocks are useful to track and of course any clocks would have still this aging component. Yeah, I mean it's not like it's completely useless. No, it's very useful. It's maybe the best thing we have actually recently, but we are still early in that process. Try to
Hmm.
develop the best biomarkers we could develop, and those biomarkers would need to be specialized for questions that we ask in a way.
Interesting.
They would
So,
not be, they would not be the same biomarker for, let's say, tracking aging and tracking rejuvenation, for example.
right. Okay, I see. So then with epigenetic clocks, for example, or you recently came up with, your lab came up with the single cell epigenetic clock where you can track the biological age of single cells, which you can probably explain why that's important over just finding out the age of bulk tissues. But my question also is that clock is looking at again epigenetic changes, right? Is there a way to train it to figure out damage, as you call it, to actually figure out whether it's this damage, aging related damage, or whether it's just epigenetic changes, some of which could be protective, as you mentioned?
Uh, at the single cell level, we don't know yet. So this is a first generation clock. So we train it based on chronological age currently. So presumably this includes aging per se, damage, and also includes neutral changes and also adaptive changes, protective changes. So it still has this aging component in my mind. So we can track, and for that reason, it works both in the direction of aging and in the rejuvenation. And we gave a few examples, but it's just I would say it's still just the first clock. We need to improve it, both just using better algorithms. Also trying to figure out how can we better track the aging per se and how we could adapt it for rejuvenation even better, and eventually integrate with other omics approaches, because I think it's a limitation just to focus on epigenetics. I mean,
Mm.
it's the best omics, yes, compared to other, but it still need to be integrated with other omics, I think.
Okay. And I don't want to glance over how this work is so transformative. So maybe you can talk a bit about why single cell clocks are so important compared to what we've had so far, which is more bulk tissue clocks.
Mhm. Well, the cell is the unit of life, right? So individual cells age. So we want to know how the cells age and how they together contribute to aging of the tissue and the organism. And for that, obviously, we need single cell clocks. So, one question, for example, could be just initial question: when you see changes in biological age by analyzing the bulk, by applying bulk clocks, why do you observe it? Is it maybe all cells equally age, or maybe 10% of cells age really, really fast and 90% don't age at all? In the bulk, you would still observe the same. Yeah. Or
Hmm.
maybe simply cell composition changes in the tissue so much that that you observe by bulk clocks. this until applying single cell clocks, it's impossible to understand. So with single cell clock, what we observe so far is that it seems like first we are kind of in the middle. First, all cells age, cells age, but at the same time they age with different rates. So it seems like some cells they age really fast. Uh, they may be unhealthy cells, some kind of senescent cells, and some cells, certain stem cells, for example, they age slower than other cells in the tissue. So in the end, we have this composite kind of measure of the tissue. But again,
Yeah.
we just developed this single cell clock, so we haven't had a chance really to apply it everywhere we could. And we only developed it for the mouse. For the human, we don't have it yet. And also, just there's not enough data sets because everybody is doing RNA-seq. It's just not enough DNA methylation kind of data sets. I wish there
Mm.
would be as many DNA methylation data sets as in the case of RNA-seq. And so we could apply even more to many contexts.
No, but that's super fascinating. I'm very excited about these new types of clocks coming up, which make the precision of even understanding rejuvenation strategies or aging much better. When I was talking to Dr. Morgan Levine, she's also working on her systems age clock, which I guess is something similar where you can measure the age of different tissues and organs as opposed to just giving you a holistic
Yeah.
organism age. So
No,
it's
but what
exciting work.
Morgan does is just, yeah, it's very important actually. Yeah, trying to understand what exactly clocks measure, how we could decompose them and develop the best clock. It's really a super work that they do. Yeah.
Yeah, no, looking forward to more work on single cell epigenetic clocks too. Um, Going back to aging, just talking about some of the theory of aging, you also have, I'm not sure if you call this the developmental theory of aging, but maybe that's what I'll go with right now.
No, we don't
You
do
don't
that.
call it that.
We
Okay,
don't do it,
never
no.
mind. You should correct me then.
Because I think uh, aging happens. I mean, aging is separate from development. Yeah. And so in a way, there are two parallel processes. There is development and aging. So we cannot say developmental kind of theory. No, I would say because it's different from development. Development is a program. Development is
Mhm.
something which we need to build an organism that would compete with other organisms and kind of give its genes to the next generation. And aging is a consequence of that. Yeah, and the aging, in my mind, happens during development and after development as well.
Yeah, and that's what I want to talk about, where you show that, contrary to popular belief, where it was previously believed that the germline, which is the reproductive cells, they don't age, but you say that, well, they actually do age, they do accumulate damage, but then there is this rejuvenation event during embryogenesis, and then again the aging kicks in, which I guess happens in the womb itself, even before we're born. So, talk a little bit about that research and what are the implications of it to understand aging?
First, prior to our research, I think there was a contradiction, and somehow people just would not discuss it or notice it. I don't know, because on one hand, you could find thousands of statements like germline does not age. It's like, so many papers like this. And of course, it's reasonable. If it would age, how could organism begin life at the same low biological age? Yeah. But then other studies, they basically say, oh, look, we can build a clock for the oocytes, so clock for sperm cells, and so it seems like these cells accumulate damage. And there's geneticists, they track mutations, epigenetic people try epigenetic changes, so it seems like the age. And okay, there's a germline ages, but then there's a statement germline does not age, and just kind of people just completely ignore this kind of contradiction.
Mhm.
So we've been thinking, so if germline ages, that means it needs to rejuvenate somewhere, but when? When can it rejuvenate? So I think basically the only way it can rejuvenate is after conception, right? So, and then we just look at like telomeres. Telomeres extended, after conception. So first
Mm.
they shorten in the germline, and then after conception, during development, especially early development, they actually extend. Yeah. But of course, other damage types is very difficult to quantify, and there are no good methods, unfortunately. So that's why we thought, okay, we need to use epigenetic clocks. Actually, it's a tool to quantify aging, right? And so when we apply it, yes, indeed, the age of the zygote, which is the fertilized egg, is not zero. So of course it makes sense because the germline aged. So now upon fertilization, biological age is not zero, but and then it decreases. It decreases to the low point. We call this point the ground zero point. And so we find it in the mouse primarily. We study that it's actually we don't know exact day, but let's say day maybe 7 or 9, around that period when it's the lowest, and
Hmm.
then it begins to increase. And so it's actually implications. There's a lot of very important implication because there's a this place during development. It's a very very special phase of development actually. So for example, many genes which are expressed at that point, they are evolutionarily old genes. These are the genes which evolved like 1 billion years ago. So they are also very conserved genes. They are much more conserved than the genes which are expressed early in development and late in development. Uh, Also, like variance in gene expression is also very low during this period. And morphologically, when you look across vertebrates, you also can hardly distinguish them. They look very alike. So to me, actually, it's not just aging begins, but we'll tell you a little bit controversial idea, but maybe the life itself, the organismal life, begins at that point.
After the ground zero point, you're saying?
Yes, the ground zero would be it's the beginning of aging and beginning of organismal life. And what is before that, like this phase of like in the mouse, first 7 to 9 days, or in the human maybe first 14 days or so. It's like maybe a preparatory phase. It's the
Hmm.
phase of damage removal, kind of and preparing the organism to start its life. Like for example, early in development, you have okay, there's a blastomere. When the zygote begins to divide, there's a blastomere. You take one blastomere and separate it, and it could become another organism. That's how twins are formed, for example. Yeah. So there's no individuality. Or you could also take these early embryos and combine them. So even
Hmm.
from different species, like forming chimera, like mouse rat chimera, for example, like this. You add, well, that's actually a very typical procedure. You just add iPS cells. Like you could add red iPS cells to mouse, blastocyst, implant it to the pregnant female, and you get a chimera composed. So there's
Hmm.
no individuality of the organism. So can we say that we
Interesting.
can say cellular life begins early, but does organismal life begin? There's no self-identity, self-recognition. There's no immune system. There's no sensory kind of systems there. You could completely separate and combine stuff, and but then at the point of extrapolation, which is like that period of ground zero, something special happens. You form, three germ layers. You generate, the primordial germ cells, and then you decrease biological age, extend telomeres, decrease damage, and you're ready to go. And so that's what I think. It's just a very special phase, and it's most rejuvenated. Aging begins there, and life begins there as well. And but then we need to understand how exactly this organism, how the zygote and dividing zygote reaches that point. What is the mechanism? How does it remove the damage? It's like so all the damage that is generated is removed. It's complete rejuvenation. It's not like the rejuvenation by Yamanaka factors or not rejuvenation like parabiosis. It's a complete and it's natural. So let's find the mechanisms, and maybe we could find something and apply this to adult cells, somatic cells, and maybe try to rejuvenate them. That's kind of the big idea.
That's nothing short of fascinating. I mean, it sounds like magic, right? Where you take these cells which have damage and then just remove it. I would, yeah, if we can understand those mechanisms, it could be so groundbreaking.
Mhm.
I have a question there, though. So then there's, I believe, emerging research coming out where, the parents' lifestyle during I mean, before conception or even just in general, their lifestyle in their youth can affect the development of the child. But if this damage is removed or cleared away, then why should that really affect the development of the child or the health of the child?
Well, what we observe is that this natural rejuvenation is efficient, but of course, damage is never a zero. We can never reach a zero damage level. Uh, Damage is, well, the simplest way to show it is maybe by mutations. So the genome is imperfect. So at any state, you have this, variants which are not good, which generate damage. So it's already like genetic damage is already non-zero, and presumably other damage forms as well. So this epigenetic imprint of the choices that the parents make, it's still reflected in their germline and later in the development in the development of the offspring. So yes, the damage is removed, but it's perhaps not 100%. It's most of the damage is removed, but some may still be left, and it may manifest itself, in the offspring's life.
I
That's
see.
the
Okay.
kind of thinking. Yeah.
Interesting. Now that explains it a bit. I'm reading Countdown by Shana Swan. She has really fascinating research on just how plastics, phthalates, and other chemicals impact just sperms and eggs health, and how that impacts the development of children
Mhm.
going forward,
Yeah.
just fertility rates. Yeah. So, in keeping with this theme of the research, you also showed how rapamycin during development, I believe it was first 45 days in mice, significantly extended lifespan and health span. Maybe you can talk a bit about that research. And I was also curious because I think it did show that the mice were smaller, so it potentially has other implications too in terms of growth.
Yeah. No, we wanted to again interrogate this relationship between aging and development. So by applying rapamycin early in life, we basically what we do is we attempt to slow down. Uh, we affect protein synthesis, yes, and uh, we slow down development, and by doing this, we slow down the aging because aging is a consequence of development in a way, yeah. So, and this trajectory kind of continues. So that was our hypothesis, and it seems to work. Um, yeah. So, but of course, our manuscript is still under review. So we just, I mean, what you mentioned is a bioarchive version of the paper. So it's now under review in a journal. But there's another group. There's an Italian group that actually have and they also submitted paper to bioarchive. We submitted back to back. Actually, they have similar findings,
Hmm.
and so they found it in mice and flies. We found this in mice and Daphnia, uh, and so it seems like kind of each study validates another study. So it seems it's true. So we are very excited about this because this also could be a method to perhaps screen other compounds because we don't need to wait until organisms die. We could quickly check during development. Of course, the delayed development could be for various reasons. It could be because we slow down development, but also because we could damage developmental genes and they just become less efficient and therefore develop slower. So again, there are two sides, but at least uh, we could still enrich for interventions that actually might extend lifespan by targeting development. So
Okay. So I guess this has possible implications for testing other compounds as well. But for say rapamycin specifically, I believe when I was talking to Matt Kabelin, he mentioned that rapamycin has beneficial effects even when you take it in middle aged or late in life. Do you think that, and this is of course going to be a hypothesis because it has been tested, but do you think that it would have higher beneficial effects or more extended lifespan if taken during development versus if taken during middle age or later in life?
Yeah, I don't know. I guess I mean, we do work on rapamycin, but I guess I don't know it intimately enough to guess what the effect would be stronger during development, middle age, or late in life. I'm not sure. So I think it just needs to be done in a well controlled study that where somebody would test all 3 points at the same time using the same model, avoiding batch effects, and then we would know. But I don't know. Maybe it would depend on the sex, depends on the concentration. There could be many covariates that kind of need to be considered.
Because I believe this had larger effects in male mice, right, compared to female mice.
In our study,
Yeah.
when we applied rapamycin during development, yes, the most of the effect is in males.
Okay. And the first 45 days in mice, what does that translate to humans then?
Uh, I don't know, 45, maybe 30 years.
Okay.
Up to 30 years, maybe. Yeah.
Yeah. Yeah. Okay. Interesting. So, just last question on this: where do you see, where's the future of this research? Like, what are the next steps you're doing as it pertains to rapamycin and development?
No, we'd like to develop it as a model to screen for longevity interventions. It's just simply because it can be done very fast, like within a month. Uh, and then maybe again, so we would not know. It's not like every intervention that delays development would extend lifespan. I don't think so, but at least it would enrich for such compounds or other manipulations, interventions. So we
Are,
think along that way. Plus, we probably need to understand mechanism a little bit better. So there's extension of the work which is being done.
are there any other compounds right now that you have in mind that you're excited to test?
Yeah, we have a number of compounds. Actually, we have a very nice program in the lab where we identify new compounds that extend lifespan. The way we identify is through kind of unbiased approach. It's kind of in silico approach in which we develop what we call longevity signatures. These are gene expression patterns, metabolite patterns that tell the potential to live long. So, for example, if we compare like across species, like across 40 species of mammals, across various interventions which are known to extend lifespan, across interventions that are known to extend lifespan for different, like a fraction of lifespan. So this that kind of effect incorporated as well. And then we generate these signatures, and then we test. Okay, does that compound change in expression in the same way we want it to be changed? And if it changes, we test first in cell culture. If it's recapitulated, we test in mice. We make a diet, give diet for a month to an animal, and if we see that it's actually changes animal in a way we think it's reasonable in terms of the extension of lifespan, we actually test it for lifespan. So we testing in old animals and also across the entire lifespan. And so we are finding new compounds this way that extend lifespan. So it's yeah, we are very excited about this project. But again, so it's still like in the middle in the middle of it. But I think it will generate new compounds that extend lifespan, and hopefully, um, some of these compounds will be tested in humans. Because in humans we already have a few candidates like rapamycin or metformin, other compounds, but I think we need more really good candidates, because eventually out of these several compounds, maybe some actually will work in humans, and this we could use actually to live longer. That would be great.
Mhm. Can you give us a sneak peek into some of the compounds, or is it too early?
It's too early because we're still working on them. So not only it's unpublished, but we're still characterizing them. Yeah.
Okay.
So we have this great collaboration with Rich Miller at University of Michigan, where we're testing these compounds, and yeah, but again, so other compounds are still being tested currently.
Yeah, I spoke to Richard Miller a few weeks ago, actually. Amazing conversation. So,
Yeah.
anyway, we'll have
Great.
to get you back to talk about some of the compounds later. um, okay, I guess one last thing. I want to talk about some rejuvenation strategies, therapies, some of which you already mentioned, the Kanpai's work or Yamanaka factors. And you wrote a paper review on this. Where you distinguish between, slowing aging is different from actually rejuvenation therapies. And you've already spoken about how you think of aging, but maybe you can talk a little bit about that distinction and how it should be viewed separately.
Well, in a simple way, one can say that we alter an organism so that it ages slower, or we alter an organism so its biological age actually is decreased. So that's the difference. That, to me, that's the rejuvenation. Sometimes in the literature, like rejuvenation is used interchangeably, and they say, well, if it's like slow in aging, actually rejuvenation. To me, it's not. Rejuvenation means a lower biological age reversal. And because we have now these tools, clocks, we could apply them and test. And apparently, there are various ways that may be possible to rejuvenate, to decrease biological age, but the mechanisms are probably different. Uh, I guess it's all started with Yamanaka approaches, because he converted somatic cells to embryonic stem cells, which are embryonic, that means young. And when we apply in other labs, of course, apply the clocks, they seem to be young. So that's good. It looks like rejuvenation. Excited a model is a parabiosis, heterochronic parabiosis. It gives a very strong rejuvenation effect,
Hmm.
observed at the level of various clocks, physiological measures, and even lifespan. So, but then it kind of raises, many other questions. Just, for example, if you have like a young blood in an old animal, what is the age? Like, how would you quantify aging? Well, in general, like you have like one young tissue in an old animal. What's the biological age of that organism, and how do they influence one another? Like this, like young tissue influences old animals, old body, and old body influences young tissue. Those questions we are also very interested in addressing. Currently addressing in the lab. Uh, this is it like some tissues may be more important than others in influencing age, or it's simply like a mass of the tissue. Let's say if you have a small tissue, it would not have much effect on the old body, but maybe a bigger tissue might have more effect. But we don't know. But maybe we can not even define then the biological age of the organism, because what's the way nature designed the organism is that things are kind of aging coordinately. But if you put like a young kidney in the old body, what's the age? I don't know. It'll be very interesting to know.